Our next speaker is Dr. Cara Wells, and she is going to talk about embryo technology and AI and some things like that. And our industry is, in fact, working. I actually just learned last night that there have been embryos and babies created from fallow deer here just recently, which is really a great, I'm thrilled to hear it. But our industry is kind of on the very, very, very beginning of where this is capable of going. And so when I met Dr. Wells, I thought, you know, while this is a little early on in the evolution of what we're doing, it's very well developed in the world of cattle. And so I would anticipate, because I'm personally working on some embryo work with bongo and with Grevy zebra, that I think that she's got a presentation that can greatly inspire, spark some interest, and spark some curiosity with how is it that embryos can be utilized as a mechanism for improving our reproduction rate within various species, particularly those that are critically endangered or are on the verge of extinction. So with that, I'd like to welcome Dr. Wells.
Hi, everyone. Thanks for having me. So I think that introduction was pretty great, but I am a reproductive physiologist turned entrepreneur. So I'm going to introduce some concepts and everything today that hopefully inspires people. And if we leave you with questions, that's fine. Just come see me afterwards. So when we talk about assisted reproductive technologies and assisted reproductive techniques, it kind of means exactly what we said. It's assisted. It doesn't happen in nature. It takes us to exert physical energy to manipulate the reproductive system. So as just an overview, before we get started, I want to talk about two very common assisted reproductive techniques, which are embryo transfer and in vitro fertilization, or IVF. And these are so important because they allow the female to maximize her genetics. You know, it's pretty easy for these males to produce multiple genetic offspring each year, but the female is typically more limited. Most non-litter bearer females, especially the large species that we're working with, they're limited to max of one offspring per female per year. So using techniques like in vitro fertilization and embryo transfer can help us multiply these females' genetics faster because we can take her embryos and put multiple embryos into surrogates so she can have genetic offspring without doing the work. So embryo transfer and in vitro fertilization, the goal of both of these is to get embryos out of them. Embryo transfer, the big difference between embryo transfer and in vitro fertilization is that in embryo transfer, the eggs or oocytes are fertilized inside of the body, and in vitro fertilization, the eggs are fertilized outside of the body. So how does this work, and how can we, set up your animals? This is a cattle example because I already had this slide made, but for embryo transfer, or what's here on the left with the superovulation, this is where the female is given a hormone treatment so her ovaries will ovulate more eggs than she would in a natural cycle. We then artificially inseminate her, and the embryos can be flushed from the uterus several days later. For in vitro fertilization, the eggs are actually aspirated directly from those ovaries, taken into a lab, fertilized in the lab, and we can grow embryos in culture. Either way, the goal is to have multiple embryos from these females, which can be transferred as individuals into surrogates of either the same species or a very closely related species. So we can do a lot when we have the ability to care and culture embryos and genetic material outside of the body. So the first advantage is what I just talked about, how we can perpetuate the genetics of superior females. This helps us reproduce these animals at a faster rate than can be achieved in nature, and it can also help us shorten the generation interval of these animals because they can be bred sometimes at a younger age. We can also freeze genetic material from these animals which can enable long-term storage of them. In terms of conservation, I think it's pretty cool that we can freeze embryos today, and in the future, those frozen embryos can be a source of genetic diversity if we're trying to use animals that, don't exist in the future, but they exist today. Embryos and other gametes, such as egg and sperm, they're very easy to ship, transport, and export versus the shipping and exporting and transferring of live animals. An embryo is about the size of a grain of sand, and that's a whole lot easier to ship across the country than an animal who stresses out in a trailer. Outside of just embryo transfer and in vitro fertilization, advancements in assisted reproductive techniques can allow for selection and control of the sex ratio. So say you want breeding bucks or breeding females and you're tired of getting that 50-50 ratio, we can do things to control this. Methods that are available today to control the sex ratio are sperm sorting. So this is a process where sperm cells are actually dyed with a fluorescent dye. They're passed through a flow cytometer machine, and because X chromosomes are bigger than Y chromosomes, the X chromosomes will absorb more dye. The machine will read this and dump the sperm cells carrying X chromosomes into a female pile and the ones carrying Y chromosomes into a male pile. Then you can artificially inseminate the females with the sex semen, and it's over 90% accurate in getting the sex of offspring of your choice. There are also some chemical diluents that we can add to semen before artificial insemination that inhibit certain sexes and therefore promote and give the desired sex sperm cells an advantage to fertilizing the egg. And we can also biopsy embryos to determine their sex, but that is invasive and you do risk damaging those embryos if we do the biopsy process. I think it was touched on it earlier. Gene editing techniques are available as a reproductive technique. We also have cloning. That is making a genetically identical copy of an animal. This can be used if we want to basically make an identical copy of that animal. Say you have an animal that was like your favorite breeding stock and he died. Well, if you cloned him, you would get a very identical animal, and typically these clones have normal fertility and live normal lives. We also have advancements in stem cell research, which we don't have time to really get into, but one of the really cool things that we can do with stem cell research is we can take a somatic body cell, so a cell from an adult organism, say cartilage, we can revert it back to stem cells, and we can turn those stem cells into sperm and egg and make new individuals. And I didn't put on this list, but I was sitting here thinking about it earlier, that there's also the opportunity to do some of these techniques with posthumous reproduction. So after an animal dies, we can actually collect their gametes or tissues for cloning and get one more chance to produce offspring out of that dead individual. So for these purposes, I believe that assisted reproductive techniques should absolutely play a role in conservation strategies. These really give us the advantage to give these animals the boost that they need to survive. But the current state of art oftentimes fails to produce live individuals. So let's take two very, very, very well-studied species, humans and domestic cattle. Probably every single person in this room knows someone who's gone through in vitro fertilization. Last year in the United States, there was over 370,000 IVF cycles, but the live birth rate, the take-home baby rate as we call it, was only 26%. Now, most people go through IVF as a treatment of infertility and not to mass-produce genetics, but still, I think 26 is incredibly low, and it shows some of the shortfalls that we have in these technologies. Let's look at cattle. We use IVF and embryo transfer in cattle because these are the healthiest specimens of their species, and we want to perpetuate their genetic traits because they grow fast, produce a lot of milk, and have other advantages that are desirable. Embryo transfer in cattle last year was 56% successful overall and about 30% successful for IVF. So I think about this, and I think that it's a scary statistic when we have these species that are so well studied. How are we ever going to get good, reliable, positive outcomes for species that we really don't know that much about? You know, in humans and cattle, we can take our temperature, measure heart rate, blood pressure at essentially any given time. Cows are pretty easy to catch. A lot of these wildlife species, catching them is difficult, and they're not just going to willingly cooperate in a squeeze chute. Also, in people and domestic livestock, we have huge databases of historical records. We know what average parameters for these species are because doctors and veterinarians have studied them for years and years. We also have the advantage in humans and these domestic livestock species of having large populations that we have the luxury of working with. Trial and error is not the most efficient process, but in these species, it does yield conclusive results. If we're talking about a species that is threatened or vulnerable or endangered, we don't have these large populations, and if we don't get these processes right the first time, we're going to lose those species. That's just matter of fact. So I'm going to talk about the northern white rhino for a second because this is kind of an interesting example. As it was said earlier, northern white rhinos have really captured a lot of media attention. They have a lot of funding in them. And so it's pretty crazy to me that any of this happened in the first place. But in 2012, I got to go to the San Diego Zoo, and at the time, they had 2 of the northern white rhinos there. In 2012, there was 5 rhinos on the planet. Now there's only 2 northern white rhinos on the planet, and they're sisters. 6 years later, I got to go back to the San Diego Zoo, and they were talking about their de-extinction project for the northern white rhino. And it was summarized on a 8.5 by 11 sheet of paper. It was a 4-step process, and this is what millions of dollars was going to do to bring back these animals. Step 1 is they were going to take cartilage, revert it into stem cells. 2, they were going to take the stem cells and turn it into sperm and egg. 3, they were going to fertilize the egg cells with the sperm cells that they made and culture the embryos. And 4, they would transfer the embryos into southern white rhinos, which is a very closely related species. Sounds easy, right? 4 steps, no big deal. Well, the problem was nobody knew how to do any of these steps in the rhino. And to their credit, they've made tremendous progress, but it was extremely experimental, and nobody had any idea to do it. The problem that I see is we have about 16,000 other species in decline that we don't know how to reproduce either. So what do? I'm a proponent that we should definitely try, but it's kind of bleak when we think of how many species are declining and how little we know about so many of them. So what kind of resources do we need to help these species survive and keep their populations increasing or at least sustained and not declining? So first off, I believe is collaboration, and I feel like that's kind of been the theme of today so far, which is really great. And this collaboration needs to be from both the public and the private sector. So that's great. We need zoos to work with these private ranches to figure out how we can keep and understand these animals. We also need collaboration between people working with domestic livestock species and the exotic wildlife species. The technologies and research that we can gain in the domestic model can be applied through translational science and used on these exotic species. And we also need participation from people in non-animal industries. We need biotech, we need engineers, we need geographers, we need all these skill sets to come forward and apply their knowledge base to help saving these species. Next, we're going to need technology and research. And as it was also said earlier today, basic research is great, but applied research, I don't want to say better, and I kind of feel like it's better. So we need to create research and technology that not only works, but it needs to be practical for use in these species. And of course, it needs to be safe. We also, obviously, need funding. And when I look at the funding problem, I don't really know anybody who says that they don't like animals. Everyone says that they love animals, and nobody wants to lose species to extinction. But the problem is, people need to be motivated to put funding into these animals, and that motivation usually comes in some form of return on that investment. And then, of course, we need animals to provide the genetics for their species and also surrogates to be the gestational carriers of new offspring that we produce. So, what do I do and why am I here? I'm a reproductive physiologist, and so when I look for ways that my work and the work of my company, EMGENISYS, can help preserve these conserved species, I need to look to my own expertise and see what can I do. So the first thing that we're going to do is we're going to identify bottlenecks that's really limiting our ability to get successful impact. And so one of the bottlenecks that I found when I first started looking into these problems in actually cattle, humans, and every species that we work with is that the way that we evaluate embryo health has not changed in over 40 years. It is common practice for veterinarians and embryologists to unknowingly transfer dead and dying embryos into the recipients or surrogates because we have no technology to identify the embryos that are not viable. This is a financial drain, it takes time, and it really makes this process inefficient. So what we've done, and I'll talk more about it in a second, is we've created a non-invasive objective technology to evaluate embryo health so we can at least make sure that when we're creating these embryos and transferring them, that every embryo transferred is alive and healthy and has a good shot of establishing a pregnancy. Most of my work has been done in the cattle model, but we've created a technology that with some refinements can theoretically be used in any mammalian species. So why is this a problem, and how did people evaluate embryo health before we come along? So about 100% of people who work with embryos evaluate embryos' health based on a morphological grading system or grading embryos based on their visual appearance. This involves looking at the embryos under a microscope and examining them for visually apparent traits like cell shape, size, color, and symmetry. This is a subjective process, and it takes years sometimes to train an embryologist. There's a lot of disagreement amongst embryologists on the quality grade of the embryo, and even senior embryologists do not always agree. For these reasons, embryologists fail to identify 20% of embryos which are non-viable at time of transfer and have no shot of ever establishing pregnancy. So how did we address this problem? Well, when I was trying to come up with solutions, I was thinking, what can an embryo tell us about itself and its biology that can lead to us detecting viability characteristics? And I was thinking that it's pretty obvious that an embryo is a growing organism. For an embryo to be alive, it needs to be able to undergo cellular divisions. It needs to go from a one-cell organism to a living, breathing, running animal in several months. And so to become that animal, these cells need to divide. They need to grow. They need to use energy, they need to have a metabolism. So I was thinking that if we could look at those cellular activity changes in real time, maybe, just maybe, we could better understand embryo health and viability. I also knew that people don't have all the time in the world to look at their embryos. So what we did is we tried to come up with a fast assessment to look at these embryos in real time. Fortunately for me, it was kind of a combination of the right technology at the right time. When I started this, advancements in imaging, computer vision, and machine learning technologies were well underway, so we could leverage these technologies to develop solutions that were not previously before possible. So when we had this idea, it was just an idea. We had to prove it out. So I partnered with a cattle veterinarian in the Texas Panhandle, Dr. Russell Killingsworth, and he started recording 30-second videos of all the embryos that came across his lab that he would transfer into recipients in his routine practice. We started correlating outcomes of the embryo videos to pregnancy outcomes to try to understand what we were working with when using these technologies. And here's kind of what we found. So I'm hoping I can play this video. There we go. So embryos do move in real time, even if we can't see it. So the image on the left is actually a video. The embryos look completely still. I realize that, but that's actually a video. Their cellular activity is not perceptible to the human eye and brain, but it's there, and we can capture this activity on a smartphone. So we use different graphic imaging processing and background subtraction techniques to make this motion humanly perceptible. So when I played that video, I don't know, it's not gonna let me play it again, but when I played that video in the middle, every time there was a white dot, that was a change from the pixel before. We can quantify this activity and compare it to pregnancy outcomes. Also, on the right, we can even detect the magnitude and direction of that motion. The ability to measure these parameters gave us never-seen-before characteristics, which we found directly related to embryo viability, embryo stress, and even embryo sex. We trained machine learning models to look for patterns in these embryos' activity over the course of that 30-second video to create predictions. To automate this, we developed a web-based platform. Today, veterinarians can record their embryos with their smartphone mounted to the microscope, which is equipment that they already own. We are not a hardware company. They record this 30-second video, upload it into our web-based platform where the machine learning artificial intelligence analysis happens behind the scenes. We scan each embryo frame by frame, searching for signs of life. We immediately send a digital report to that veterinarian or embryologist, and they can use this objective data to manage their breeding strategy. And as we got deeper into these studies, we found that studying their activity in real time could do more than just predict embryo viability. We found that we could detect stress subclinically. So, for example, we did a study where we brought embryos from body temperature, which is where they're happy, to cooler ambient room temperature environments. We saw as the embryos got cooler, they actually sped up. To us, that was the embryo showing signs of stress. If we see a stressed embryo in real time, the lab group can take strategies to mitigate that stress. They can alter the culture media, alter the temperature, and do things to make that embryo happy and grow again faster than waiting the embryo to die and then, just being frustrated that they had another loss. We also saw that their activity correlated to embryo sex. Male embryos grow faster, and we could see this even in our short 30-second real-time analysis. So with this data, we feel like we can create answers faster than what was previously possible. Many of these exotic wildlife species, which was said earlier, are very rarely studied. We know that in theory, these assisted reproductive techniques can be critical strategies to their species survival, but right now, we don't even know how to grow and culture a lot of these embryos. We don't know if the hormonal stimulations that we're using creates quality embryos. I'm not saying that my technology has all the answers per se, but we can definitely help provide objective data to get to these answers faster. And with the imminent demand to save species from extinction and decline, we need fast, reliable solutions. We need to reduce trial and error so we can create solutions to transfer viable embryos and create protocols which are conducive to their growth, survival. So how can you help and how can we collaborate? I know a lot of these concepts may not be something that you're working with today, but I am definitely willing to be a source of information for you if this is something that you're willing to try in the future. And if you're already using assisted reproductive techniques in your wildlife species, I would love to work with you, collect videos with you, and see what we can see with our technology. I'm personally pretty passionate about applying these technologies to wildlife species, but I don't have the resources to do this myself. We need animals, we need to build a database, and if you come and talk to me, if this is something that we can potentially work together on, I think that collaboration is the only way that we can create these models to be applicable to all these species. All this work will help us pursue our mission to improve animal reproduction and promote sustainability and welfare through advancements in assisted reproductive technologies. To date, we've trained our models on over 5,000 videos of embryos. Most of these embryos, like I said, are from cattle species, but currently these models have 85 to 95% prediction accuracy. I have about 35 veterinary groups across North America using this technology. And as a company, we've been pretty aggressive about protecting our intellectual property. So on Tuesday, we will officially have 8 issued United States patents with 7 patent applications still pending in the United States and about 40 patent applications filed worldwide. In this patent portfolio, we have video analysis of embryos recorded at speeds of 2 frames per second or faster. And we also have protected analysis of video recordings which are 10 minutes in duration or less, giving my company protection on the fastest real-time solution to evaluate embryo health in the marketplace. All in all, I think that we can create 360 degrees of impact that promotes animal health, human health, and environmental health. When we get animals pregnant the first time, it reduces cost, inputs, and time into producing these animals. My team is a very passionate and diverse team of animal scientists like myself, veterinarians, machine learning experts, and intellectual property specialists. So please, wildlife partners, join me in our efforts to promote conservation because together we are breeding something epic. Thank you.
Thank you, Cara. As I've said, my hope in all of this is to inspire people to think outside the box and to reconsider maybe the way that we've looked at ourselves and the way that we identify ourselves. And one of the thoughts I had as Cara was talking is that when you look at the exotic wildlife industry, we have a message that's being shared nationally and even globally. And the question is, what is that message? What is it that we're telling the world about who we are? And one of the consistent themes that I saw when I first got started doing this was that our primary message was that we're conservationists because we're breeding and managing species through hunting. And I'm a hunter. I love to hunt. I hunted all over the place. And I view myself as a hunter. I don't necessarily view myself as a conservationist because I'm a hunter. I really look at hunting as something that is totally different. It's an instinctive behavior that I choose to engage in. But the fact is that if you look at our country and you look at the world, it's a very, very small portion of our population that's engaging in that activity. And that number is getting smaller as the years go by. And so when we share our message with the world that in Texas we have this conservation model around raising exotic wildlife, and the foundation of that is that we're hunting the species, and therefore that's why we're significant, and that's why we're meaningful, and that's why we're valuable. I think if you look at the perception that we're getting as feedback, that's not working very well. It's not terribly effective in communicating our message. And so I look at zoos throughout the United States, and most of these large city zoos, they're nonprofit organizations, they're government funded, but they're doing things that we're not. They're investing significant amount of money in doing research like what Cara talked about. They're spending a huge amount of money that is coming to them through donations to study things like sparrows on Kauai. And we have this incredible opportunity in front of us as an industry to start getting involved in things that it tells a little different story. It's not to say the story that we've been telling for all these decades didn't work or it wasn't effective, but when we can start telling the story of, like San Diego Zoo as an example that's done all this work with northern white rhinos, when we start talking about how we've done all this great detailed genetic work with Grevy zebras, and then we went out and we started creating embryos with Grevy zebras, and we started putting them in donkeys. And suddenly we went from 3,000 Grevy zebras worldwide to 20,000 Grevy zebras worldwide. And most of them are in Texas. This has not happened yet, but it's going to happen. And when that becomes a part of our story globally, and it becomes a part of our message, we take away the weapon that organizations like PETA and HSUS and all these other money-raising organizations are using against us. When we can stand in front of the public eye and say, look at all these things that we're doing as Texas landowners, as people that are doing this from an investment standpoint. Yes, we hunt, and yes, it's a part of the model, and yes, it's a part of the industry, but here are all these other things that we're doing also. And so by bringing people like Cara here to talk about these potential technologies, and Dr. Durr to talk about the genetic work that's available in front of us, I think that I would like to encourage you to ask yourself, can we present to our society, to the public, can we present to the zoo community throughout the United States? Because at the end of the day, that's where we got all these animals, is from zoos. They just don't admit it. They don't claim us as their bastard children, but the truth is that's what we are. Our animals came from zoos. And so why don't they claim us? And they don't claim us because our message isn't great. And these kinds of things, to me, give us the ability to shape a message around what we're doing that is more inviting and it's more inclusive, and it's going to position us to not only make a better investment for ourselves, but it really allows us to wear this label of conservationist in a way that's different than the way that we've worn it in the past. And so I hope these topics encourage you to think a little outside the box and encourage you to think a little differently about what we've been doing, and more importantly, about where we're going.